everyone, welcome back to my channel. And today we are going to learn a very interesting topic in biology, which is synapse. So, what is synapse? As you can see over here, it's basically the transfer of nerve impulses from one neuron to another one. So let's go into the detail of the synapse. First of all, what is synapse? We are going to learn with the help of diagram the structure of synapse and then the role of neurotransmitters and how do they transmit the messages, and last one, the role of synapse or the function of synapses we are going to learn. So number one, what is synapse? So basically, it's a gap in between the two neurons. So it is a point where axon of one neuron connects with the dendrite of the another neuron with an effector. So it is a connection point, first of all. It links different neurons together. And number four, the human brain, we only the central nervous system and the brain has 10 exponent 11 neurons and more synapses, which are 10 exponent 14 synapses. Means further on we are going to discuss there are divergence one and the convergence one. So we are going to discuss what is synapse and how to they act. So first of all, the structure of chemical synapse, as you can see over here. Let's name the two neurons. So let's say this one is the first neuron, so we call it a presynaptic neuron. Okay, so it's a axon, and this one is, you can say, the roots from where the messages are going to conduct. So this one is the presynaptic one. Keep in mind that there is an active transport in the synapse. It has been discussed so many times. So active transport needs energy, and that energy comes from the mitochondria. So it must have lots of mitochondria to gives energy for the nerve impulse or the conduction of nerve impulse. These are the vesicles which you can see in blue, and this one is the postsynaptic neuron, and these pink one are the receptor cells which are going to accept the neuron. So this is a general structure. Let's go into the detail of the process, that how does it happen. Let's just start from here. Okay, these one are the calcium pumps. So first of all, the calcium pumps release calcium ions. These actually causes these vesicles to fuse with this membrane. So first of all, this membrane is closed. When the vesicles come near to this membrane, then they fuse with it, and with the help of exocytosis, means the material is excreted out into the external environment. The exocytosis, the neurotransmitters are released into the space. Now, this space or the connection point between the, so there is no physical connection, but it's only a spatial connection between the two neurons. This is known as synapse. So the neurotransmitters are released into the synapse, and then further they are accepted by the receptors that are present on the postsynaptic neuron and further. Okay, so when all of this is happening, the sodium ions are also released that are that causes some change in the potential, and this is how a nerve impulse is conducted. Let's go into the detail of this one. So first of all, we say that these calcium knobs, or the synaptic knobs, these are the synaptic knobs, and calcium enter through these synaptic knobs, causing the vesicles to fuse with the membrane. So the calcium ions causes postsynaptic vesicles to fuse with the membrane of um, presynaptic membrane. So when this is released, the neurotransmitters, which are basically acetylcholine, is released into the space. So these are the neurotransmitters. Then further on, when acetylcholine is released into the space, these are accepted by the receptors. Now, when we talk about receptors, whether they are related to antibody or any neuron, we must know that they have a complementary shape to the stimuli or to the whatever is receiving. So when we talk about the shape, it's about the chemical bonding and not the physical shape. So the shape, when we say the shape fits in, it means there is more chance of chemical bonding between the two compounds. Okay, so acetylcholine molecule fuses with receptor sites, and then further it causes the sodium channels to open, allow sodium ions to diffuse in rapidly along a concentration gradient. So what happened next? Due to this sodium ions, there is a new action potential in the postsynaptic neurons. So what is action potential? It is due to the change in the charges. For example, we are saying that the sodium ions are moving in. So it means that there is a difference in a charge. The charge, there is more positive charge now. So this is the active action, or we can only say the action potential, and this is called as excitatory postsynaptic potential. Then what happened to acetylcholine? The acetylcholine basically is hydrolyzed into to choline and ethanoic acid using an enzyme called as acetylcholinesterase. So it, you must know what are esters. You might have read them in um, the organic chemistry. So further on, when the hydrolysis takes place, then ATP is released by mitochondria, and it combines again the choline and the ethanoic acid into acetylcholine. So this is called as recycling. And further on, these sodium channels closes when there is no acetylcholine, and the messages are conducting to the next neuron.